Good afternoon. I'm Darcy Gentleman. Welcome to this news briefing from the 254th National Meeting and Exposition of the American Chemical Society here in Washington, D.C. We're joined today by Dr. Silvana Andreescu from Clarkson University in Potsdam, New York. She has been developing a paper-based sensor with wide-ranging applications, including telling when food and makeup have gone bad. Dr. Andreescu?
Thank you. Good afternoon, everyone, and thank you for having me here today. So we have developed a technology that can measure different components in food, different chemical markers or quality indicators in food. Our sensors look like little dots on paper, and most of the work that we do is really to engineer these little dots, the paper, with chemical responsive materials that will change color if the target is present. So for most of the platforms or the most developed platform that we have measures antioxidants in tea or wine or cosmetic products. So we can establish a fingerprint that is indicative of the composition of this sample. So if we are to think about wine, every wine has a particular makeup of antioxidants, and we measure this as a color change. So we have a code that's a little bit different than just the label of production or the date of production or the date of expiration. So we actually measure the chemical composition that may degrade over time. So we use this as a quality indicator. Other sensors we are for pesticides or mycotoxins, so things that are toxic. We look at both harmful and beneficial components. We can fabricate them in a multiplex format, so we can have a little dot for antioxidants, we can have another dot for pesticides, so that will tell us whether this is the sample is toxic or not. We have a couple of generations of these sensors. So as I said, the most advanced one is the antioxidant. The second one is for biomarkers that indicate spoilage, so markers for fresheners. And we hope in our next 5 years plan to incorporate this technology in smart packaging. Thank you.
All right. Well, thank you very much for that introduction. Are there any questions? And please remember to state your name and affiliation before asking your question. Here in the third row.
So it's Kath O'Driscoll, and it's from Chemistry and Industry magazine. You mentioned in the paper that there have been other paper-based sensors before. And I'm just curious to find out a little bit more about why your technology is so novel. You mentioned the use of nanotechnology, and I'm curious as to why you've hit on nanotechnology. What's special about the nanotechnology that allows you to do these kind of applications? Thanks.
Okay, thank you. So our sensors contain redox-active nanoparticles that are optically responsive. We also interface these particles with enzymes or DNA that provide selectivity for a particular target. So these particles essentially change color by themselves. Most other technologies that are based on paper in the literature or used by others are using soluble organic dyes. So you have solutions migrating through paper. Here we have it's a solid platform. It's essentially a spot test. We only add a sample. The way this works, we add the sample, and within a couple of seconds or minutes, depending on what we are measuring, we see a color change. Everything is incorporated within that spot, so there are no channels that need to meet or reagents. We don't. Everything is integrated, so it's an all-in-one platform. And it's very stable, if I may add. We don't have the storage problems with soluble dyes.
Is it use once and then throw away these devices?
We can look at them as a pregnancy test strip. And we can dispose them after they are used. They are very cheap. If we are, as we move further for the packaging, I think we have to be concerned, and we will look for potential toxicity or side reactions. But right now, it's as any device or the glucose meter that people use to check glycemia at home. That is the same concept, it's just that now we see a color change.
And I was curious about, you mentioned as well, consumers actually using these things perhaps to test for food spoilage in a home, whether something's gone off. I mean, could this be something, you said about incorporating in the packaging, but could consumers also theoretically have like a pack of these things that they could test for themselves at home?
We hope so.
Whether things have gone off?
Yes, absolutely. We hope so. It's very simple to use. As I said, it's simply measuring, putting a couple of very small amount of sample onto the surface of the sensor, and then there is a color change. You don't have to do anything else. So there is potential to be used by consumers as well. The technology is ready, at least for the antioxidants. For the others, food spoilage, we are still working on it.
What are these redox active nanoparticles? Are they used in other applications, in other industries? And how many of them would you use, or how many reagents do you use typically for some of these tests?
We produce them in-house. You can also buy them. These are metal oxide particles. Other people are using them. There is a whole area of research for therapeutic purposes as drugs, antioxidant drugs. There is also another area in the electronics industry where they use it as polishers. So we found a more unique application as detection probes.
Thank you.
All right, thank you. Other questions?
Katie Cottingham, American Chemical Society. So I'm wondering, would the sensor be able to kind of just automatically measure whether a product is fresh, or do you have to open up the product and then put the little spot of it on the surface?
The sensors that we have right now, we do need to add the product on the sensor. We are now working on, I have a couple of students who are working on investigating whether we can put it in a sort of a packaging and measure the chemicals that come out. So this is the ultimate target, but the ones that we have at this moment, we do need to put a sample. So if you look at wine, you do need to put a drop of wine there, or tea.
Christine Saw, American Chemical Society. So I'm wondering, what kinds of foods are you testing? Would consumers be able to use it to test their chicken, for example, like more solid type foods rather than wines and teas and that kind of thing, and liquid samples that you could easily drop onto the spot? What would you do with a solid sample?
Okay, so the most extensive study we've done was with tea. We do have now in the lab a couple of samples of solid food, and we let them degrade, and we are hoping to see the color change over time. So we essentially put the sensor on top, and some compounds that, the biogenic amines, for example, they will come out, and it's characteristic of, so more volatile compounds, essentially. We are working towards that goal.
All right, thank you. Other questions?
I just wonder, what concentrations of spoilage are you looking at? What concentrations of chemicals are being detected in order to produce this color change? That's
an excellent question, and we don't have the answer yet. The most difficult part when we develop the technology is to match the calibration curve with the concentration of the species that are coming out. So we are testing different chemistry and see at what point we can measure and then sort of go backwards and see how much is produced.
And how much roughly do you think these sensor devices will cost? How easy are they to manufacture? And
we can manufacture them by printing, so it's already, we can do roll-to-roll, we can do mass production. The cost varies depending on what we measure. If we measure simply antioxidants, then this is cheap because we don't have any additional biological reagents that are used. As we move towards more complex matrices, if we look for toxicity, we do use enzymes or DNA, which we incorporate in the sensors, and then the price is essentially associated with the price of the biologicals, but not the sensor or the nanostructure itself.
And you also mentioned multiplexing, i.e. testing different substances. How many different substances can you test for at once?
4 or 5 easily, we think. For example, if you look at fermentation, you can measure glucose, and then you can measure ethanol produced, and you can measure both increases and decreases in certain markers. So we believe we can build the platform to fit to certain needs. We are looking for collaborators in the food industry. So if anybody is interested, we will be more than happy to expand or multiplex to whatever makes more sense from their perspective.
Thank you.
I'm going to take moderator's prerogative and just ask a quick question. So when someone's using this at home, would you have a guideline that would say if the spot is this color, it means this, or would it require some other companion device to tell them what the sensor means?
We will provide the test sensor, and then we will provide a sensor with a calibrated sensor that essentially shows what color would you expect for this particular concentration. So then you can match the color of our calibration spot with the sample.
Oh, that's great. All right. Any other questions, either from the room or online? Absolutely.
Felix Roman from the University of Puerto Rico. Are you concerned about leaching some of these nanoparticles into food? Are they non-toxic?
We have tested leaching. They do not leach. We do use polymers to embed. We embed these particles. They are not only simply attached. There are a lot of concerns with toxicity of particles, and that's something that we will have to look as we move towards more of the packaging applications. If you use them at home, I think it's more, you don't ingest them. It's essentially like a test. The pregnancy test, if they contain nanoparticles, we don't really think of it as being something toxic. But these particular particles I mentioned, people are interested in exploring them as therapeutic. So there are a couple of publications there where they say they can be used to fight Alzheimer, and we can inject them and they can be drugs. So as we move further with this research, this is definitely a question we'll have to answer. But there are both negatives and then also positives, so they may actually be very good candidates.
Thank you.
Thank you.
All right. Thank you very much. Any other questions online? All right. Well, with that, thank you very much, everyone, for attending. The archive version of this session will soon be posted at bit.ly/ACS live_DC. That's capital A, capital C, capital S, capital L, lowercase IVE _capital D, capital C. Please join us for our next press conference at 3 p.m. today on mining waste from avocados for new medicines and other useful compounds. Thank you very much.